Welcome to today's PSW COVID-19 clinical webinar. We are continuing this webinar offering. We had offered a webinar on early clinical updates a few weeks ago, and we're so thrilled to have back our speakers from that prior webinar, Dr. Tom Dilworth and Dr. Lucas Schulz, to present some updates and information and recap some information for all of you today. They did review questions that were submitted in advance, and we'll try to answer those throughout their presentation. And we'll look forward to responding to additional questions at the end. And I just want to first say, I should have introduced myself. This is Sarah Sorum, PSW CEO, and just want to really compliment the work of our speakers today on being so up for last minute adaptability and offering these really timely updates. So thanks to our speakers. And then I also just want to say thanks to all of you who are really working to care for your patients at the front lines. And there's been no more time where pharmacy professionals are more essential than they are right now. We need amazing pharmacy professionals caring for patients, both in the situation of COVID-19, but also for their chronic conditions and care of populations, et cetera. Today we'll focus on the care of patients with COVID-19 and the care of populations and prevention. But I just want to applaud all of you for all the great work that you're doing. So with that, I'm going to turn it over to Erica Martin on the PSW team to provide some instructions about today's webinar, and then we'll turn it over to our speakers.
Hi everyone, welcome to the webinar. Today we will have a presentation, and then as the presentation is going on or during the question and answer portion, if you'd like to submit your questions into the question box, I will record those down. And to try to get to as many questions as possible, we'll try to group questions. So like questions may be rephrased or grouped together, but please know we're recording all the questions so that even if you don't hear read word for word, we are recognizing your question. So please just enter those into the question box and we will get those down for our speakers.
All right, thank you. So welcome. My name is Tom Dilworth. This is PSW COVID-19 Clinical Webinar Part 2. I'm very excited to be presenting again to you today and excited to be presenting with Luke from UW. Erica, can you let me advance?
Perhaps it might work now. No luck? Oh, there we go.
Okay. So keeping with the theme of last time and having a basketball picture, Luke and I are really hopeful that we can, like Michael Jordan in this picture, dunk a ball from a free throw line over all the COVID misinformation, because there is a ton of it, and we'll do our best to dispel that and give you something actionable to take away. So we'll review the current and future state of the pandemic, discuss considerations, limitations, and updates related to both testing and treatment. We hope we can recognize some lessons learned and future challenges for the pharmacy profession. I think we've all learned a lot during the last couple months and faced a lot of challenges head on, which Sarah alluded to at the outset. And then we definitely want to leave time for audience questions. So we've got 30 minutes at the end for that. So I want to start out with an update. So last time I spoke to you, I said coronavirus in the United States has the potential to kill more people than we lost in the Vietnam War. That has now unfortunately happened. It's sobering to think, but on April 28th, that did occur. It was over 60,000 deaths in the U.S. and counting. There are some projections suggesting over 100,000 by June 1st. This is an updated slide again from last time. This is the dashboard put together by the group at Johns Hopkins. As you can see on the left-hand side, highlighted in blue, we've got over 1 million infections here in the States. And then the columns on the right-hand side, you'll see 67,000 plus deaths in the United States, almost 20,000 in New York City. So unless you haven't been paying attention, New York City is the epicenter for a COVID-19 infection in the world right now. They're really hurting. Crude mortality, when you look at these numbers, is 5.8%. It's likely lower than that because testing capacity is suboptimal, and we don't know how many people have actually been infected. These are really just the sickest of the sick patients coming into the hospitals that are getting tested. One thing that I really wanted to highlight, because I think it's important, is this pandemic has amplified health disparities in this country. I think we're all aware that people of lower socioeconomic status and means, people with lower education levels, people of color often suffer worse from chronic diseases and infectious diseases than people that don't meet those criteria. And this was an interesting viewpoint published in JAMA, and it showed the proportion of a location that was African American and the proportion of COVID-related deaths in that location that were who were African American. So as you can see, Chicago, 32% of the city is African American, 70% of those who died were African American. Same story in Michigan and Louisiana, but they actually called out Milwaukee in this viewpoint. So Milwaukee, biggest city in Wisconsin, 26% African American, 81% of the deaths in Milwaukee at the time of this viewpoint being published, 81% of the deaths were African American. So, when we talk about opportunities and challenges for our profession and for our healthcare enterprise at large, we definitely have an opportunity here to look ourselves in the mirror and address some fundamental disparities in care and outcomes. And the question that I will kind of leave you with is, will we do that? Or what can we do to alleviate some of those disparities? Keeping on with the data, you'll see here that we are the epicenter of COVID worldwide, far and above any other country. When you look at Wisconsin, it is a bit of a different story. Now, we were an early adopter of school closures. Governor Evers did that before he enacted a safer at home, and that is almost a de facto safer at home for a lot of people because if the kids aren't in school, then it's hard for parents to go to school, and then we've done our safer at home. And as you can see, when you talk about flattening the curve, it's flattened, and it's not completely flat, but it's flattened nicely here in Wisconsin. Now, last time I talked about a surge prediction, there was a group out of the University of Washington that had a nice website, interactive, suggesting, when each state would hit its surge in terms of COVID and what this impact would be on hospital beds and ICU beds and ventilators, et cetera. And Wisconsin was predicted to be just about last week. We actually peaked a little bit earlier than that. But what's very interesting, so this is the State Journal on April 15th. So we've definitely peaked. The surge has not been as large as I think anyone anticipated it would be in the state of Wisconsin. But what is notable is that we're sort of at a steady state right now. So we've gotten our peak. It wasn't what we expected, but we are not seeing a downswing yet. So, I sit on multiple calls with my colleagues at Advocate Aurora throughout the week. And, the consistent refrain the last 2 weeks or so has been it's status quo. We don't see more infections. We're not really seeing less infections. It's just sort of status quo. So that's kind of what I would say about where we're at right now is we're at a steady state level. But the peak was not as large as we thought it could be. So now I want to talk a little bit about the Badger bounce back because this comes up a lot, both from patients and then people in my orbit and people that I work with. What does this mean? Well, we're still in safer at home. Safer at home was extended till May 26th. And now, consistent with recommendations from multiple societies, and I'll share those in a second, they're developing objective criteria to gradually re-enter, I'll say in quotations, normal life without social distancing. There's gating criteria and there's core responsibilities. So you have to see downward trajectory in influenza illnesses for 14 days. We're definitely seeing that. We're out of peak seasonal flu. Downward trajectory of COVID-19 like cases, the different signs and symptoms, and then a downward trajectory of positive tests as a percent of total tests. And that's really, testing capacity is going to be big there. And that's part of the core responsibilities that every Wisconsin resident who has symptoms can get a lab test that's not the case right now. And then in terms of tracing, the Wisconsin DHS is hoping to hire 1,000 people per their website to help trace. I don't know where they're at in that hiring, but I would guess they're not nearly close to 1,000. And this is just a graphical depiction of two of those gating criteria. So on the left-hand side, you'll see number of ED visits with suspected COVID-related concerns, and then the trend line going through May 3rd, and the trend line is slightly up, so it's not going down. And then when you look at people tested for COVID with positive results, you see, like I was alluding to, basically a steady state. So both of these analyses indicate there's been no downward trend in the last 14 days. Now, if this was May 25th, and that's what these data look like, the way the bounce back is written, we would still stay in safer at home. We would not enter into phase one, which would allow gatherings of up to 10 people, and public schools could reopen. So I would encourage you to take a look at the bounce back, because it's very informative. One other thing that's been highlighted by DHS and the governor's office is our use of safer at home and social distancing in Wisconsin is estimated to save 300 lives. I do want to reiterate the importance of social distancing. This is basically all we got right now, guys. So as a pharmacist, you have to relay the importance of social distancing to everyone in your orbit, be that family, friends, patients, colleagues, whomever. Use social media, reinforce this. There's a lot of misinformation right now about the need to preserve individual liberty and reopen the economy. And there is a give and take here, but we're not ready, okay? Because we don't have testing capacity that we need. We don't have treatment, and Luke's going to talk extensively about the potential treatments, and we don't have a vaccine. So really our only treatment right now is prevention. And until we get some of these other things in place, we're going to have to rely on prevention and distance. Down on the bottom left, you'll see a letter that I was very happy to be one of the members of the Society of Infectious Disease Pharmacists to help sign on to this with IDSA and American College of Clinical Pharmacy, which is basically urging federal, state, and local governments to prioritize safety with their COVID-19 policies. And by that, using objective data to guide the continued use or discontinuation of social distancing practices versus just emotional reactions to the public saying, we're sick of this and we'd like to, go back to the stores. If the data don't suggest that's reasonable, then we're saying you shouldn't do that. And that's what that letter argued for. These are data out of Wuhan, China, where this whole pandemic started. And I think they're informative because a lot of people, they say, well, if I go back a couple slides and I show you this, this graphical representation just says, it's sort of a hypothetical. This is what's going to happen if we don't do anything, and this is what will happen if we do something. But let's actually put some numbers on it. So if you look at the number of cases in Wuhan of coronavirus, COVID-19, you'll see different temporal periods on the bottom, and you'll see the number of cases on the y-axis. So as you see, the start of the Chinese New Year, or the massive migration, led to an explosion in cases. And they did city lockdowns, traffic suspensions, and home quarantines. Really wasn't enough. Then they started cohorting patients into different groups using centralized quarantine schemings. They better understood treatment and medical care and how to prevent the spread of it to healthcare workers and within the hospital. And then eventually they started screening everyone in their community for symptoms and isolating people, and they've expanded testing, and they've got their case numbers down. Another way to look at this is how well the virus spreads, the reproduction number. So as you can see, the reproduction number estimates on the y-axis and the time on the x-axis, and you see the different sort of iterations of social distancing that went on. Now, obviously, this led to a reduction of a transmission reproductive number less than 1, which I'm going to talk about on the next slide, what that means. But I do want to reinforce one thing about these data, and that is the measures that were implemented from a social distancing standpoint in Wuhan are much, much more draconian than what's going on right now. I mean, these people were forced to stay in their house, sometimes 6 days a week, and they'd be like a day a week they were allowed to go out and get something from the grocery store. So, we're sort of in a pseudo social distancing. If I were to put put a name on it, because we're still sort of allowed to freely leave our homes, and we can go to the home improvement store, and we can go to the grocery store, et cetera. So, this was much more draconian, but it did achieve the results that were intended. When you talk about reproduction number, I do think it's important to just kind of review what this means. It's basically a measure of how contagious an infectious agent is, and it represents the average number of people that one person with a virus will infect. The sort of worst case scenario is measles. So measles has an R naught of 12. Every person with measles can affect 12 people. Most other viruses aren't that high. But as you see here on the right-hand side, COVID is at about a 2 to a 2.5. New England Journal cited it as a 2.2. Ebola was 1.6 to 2. H1N1 flu a while back was 1.2 to 1.6. So you get some sense of comparison. The MERS coronavirus, which was sort of the coronavirus that afflicted the Middle East a while back, was upwards to 7. So if you have a R naught less than 1, the disease is going to die out. If you have R naught equal to 1, and I missed the 1 there on that second bullet, the disease stays present, but you won't have a pandemic. And if your R naught goes above 1, your disease transmission increases, and you have the potential for a pandemic. So that's what we're trying to avoid. I want to briefly review lab testing. Not much has changed in terms of lab testing. It is still a rapid transcriptase, polymerase chain reaction, or real-time PCR. False negatives are possible, but they're not as widespread as it was thought initially. There was some initial data out of China looking at comparisons of PCRs versus chest radiographs, and suggesting that the sensitivity of the PCR was very low, in the 70% range. It's hard to put a great number on sensitivity because the virus is new, the tests are new, they're emergency use authorization, they're not FDA approved. There's not a lot of great comparative data. But if you do a good collection, a good nasopharyngeal swab, and it's not immediately after the person was infected, which is sometimes going to be obviously difficult to know, the sensitivity is very high. Now, you can get a false negative, like I said, in early asymptomatic infection, or if the specimen is collected improperly or mishandled. If that's the case, and you've got a potential false negative, but it's a person of interest, the CDC does recommend retesting multiple respiratory sites, including maybe a BAL. But again, the sensitivity is pretty good for this test. So don't believe the data that were coming out of the medical literature initially. Now this leads us to the next phase of testing here, and that is pharmacists and testing. So what's remarkable is, not remarkable, I shouldn't say it's remarkable. What's exciting is that the Department of Health and Human Services said on April 8th that pharmacists can now test for COVID-19. And as we know, we are the most accessible healthcare provider. There's pharmacies all over this country. So there's a potential to assess community spread, which would be a big boon for our understanding of this virus here in the United States, because that's what we don't know, right? I showed you 1 million infections and 70-ish thousand deaths and a crude mortality rate of 5.8%, but I said that's likely an overestimation because we don't know how many people in the community are affected because they're not seeking care. Well, pharmacists could be doing some of this testing that would help us understand the community spread. However, and PSW is out in front of this. They had a great statement that I've copied some of the highlights here. But there are logistics that need to be addressed before we can, as a profession, step headfirst into this. The first is PPE. PPE continues to be a precious resource, but you need PPE if you're doing these tests. There's also questions about, how are these tests going to get ordered? What are the limitations of the tests? There's potential for false negatives. Like I said, it's uncommon, but it can happen. And then to whom do these results get reported to? To the physician, to the public health authorities, and how does that reporting happen? Is there an electronic scheme that can be set up? So there's a lot of logistics that are going to have to be put in place before this can be widespread throughout the profession. Again, ACCP, SHEA, IDSA, SIEP, and a number of other groups put together actually a response to DHHS and said, here's a number of things that you need to do and pursue these recommendations before pharmacists can readily engage in testing. And then when you talk about testing, you have to talk about serology or antibody testing, because this has really been a big issue, particularly in the lay press. There was a number of preprint studies, so studies that were undertaken by researchers. They were published online without peer review, so everyone could read them. There's pros and cons to preprint literature. We could do a whole seminar on that, and I'm not going to go into that today. But I've cited one of them here. This was Santa Clara County, California. And this study and a couple of other studies, one was out of UCLA, generated a lot of criticism. So there were some who used these results as fodder to argue against the need for social distancing, because what they found was what you would expect to find. You find high rates of COVID-19 seropositivity in the community, suggesting the virus is much more widespread than we thought. And then if you look at the number of people who have died, it's actually a much lower proportion of patients who are infected. And the crude mortality rate is potentially akin to seasonal influenza. And we'll talk about why that's not the case here in a second. But again, these studies were heavily criticized. They were non-random samples. It's hard to do a random sample for an antibody study, but like the Santa Clara study, they enrolled through Facebook. So you had to be a Facebook user, which I know a lot of people are, but it's not everyone. So the non-random sampling does limit its generalizability. There's also different tests used in these studies, and test accuracy has really been called into question. You can get false positives of other endemic coronaviruses that are not SARS-CoV-2. I think the bottom line here is many more studies are coming. You could even be asked to enroll in one of these studies in your community if it comes that way. So I think there's going to be a lot of work and research in this area. It's going to be a big part of our public health armamentarium going forward with COVID-19 understanding and containment is serology. But I do want to talk briefly about this notion that seasonal influenza and COVID-19 are similar. First of all, to go to the reproduction number, it's much higher for COVID-19. And then when you look at mortality, CDC estimates 24 to 62,000 deaths from seasonal flu from October of 2019 to April of 2020. And I just showed you since April of 2020, we've had over 67,000 deaths from COVID. So, you don't have to be an expert. You can look at New York City and you can look at Northern Italy and you can look at Spain and you can say this is not the seasonal flu. But again, people, particularly journalists, they look at these preprint studies, and typically when studies are published in peer-reviewed journals, they're embargoed, they're peer-reviewed. The journalists have time to review the data, talk to the authors, talk to other experts, and come out with a good message to the lay public. But when they're put up preprint, none of that happens, and the journalists are sort of left to their own devices to opine on the data and what they mean. When you unmoor data from the context you need to understand it, you can get some erroneous conclusions and erroneous narratives, and that, unfortunately, I believe is what's happening right now with the serology narrative that's being used to argue against the need for social distancing. When you talk about serology, it's just not ready for prime time. We don't know the immunologic correlates for SARS-CoV-2 protection, or the proportion of a population who must achieve them. We also don't know if antibodies to SARS-CoV-2, the spike protein, lead to functional and or durable protection. Now, when you look at SARS-CoV-1, the previous SARS pandemic, people who developed antibodies, those antibodies did persist for over a year. But you could make that leap for this current virus, but we don't know. The virus hasn't even been around for a year. There's also the window period where someone needs to develop antibodies. So up to 20% of symptomatic SARS-CoV-2 patients have little or no detectable antibody. We do know that the SARS-CoV-1 survivors had antibody, but we don't really know about the non-hospitalized SARS patients, and we don't really know about that to a certain extent with SARS-CoV-2. Now, if you were to try to guess what level of community protective immunity or herd immunity, or what level of protection we need in a community to achieve a herd immunity, with an R naught of 2.2, if you look at measles as an example, which is this graph here, you need about 60%. And I don't think we're at 60% seropositivity in the community. And I don't know how long it's going to take us to get there to even understand whether or not we're at 60%. So I'm just trying to give you an overall sense of this, that it's going to be really important to understand these serology data, but it's not quite ready for prime time in terms of making decisions that affect public health. So on that line, people often ask me, well, did we overreact? And I keep going back to Anthony Fauci, who's the physician advising President President Trump as it relates to COVID-19. And he said months ago, it looks like you're overreacting. You're probably doing the right thing. And that's what I keep telling people. I'm like, yes, it doesn't feel right. And it's, this is not how anyone wants to live their life. But if you're overreacting, you're doing the right thing. I looked at Germany as a very good example of this. So Germany has, as of earlier last month, relaxed some of their social distancing restrictions. They let people back into public places like zoos, and they let their children back into schools. And in mid-April, their reproduction number was 0.7. Again, you want it to be less than 1. Then as April progressed, they got to 0.9. And in late April, they were at 1. And then Angela Merkel, the Chancellor of Germany, was on record saying, look, I've conferred with the epidemiologists and the public health officials. If we get to 1.1 for an R naught, our health system is going to max out in October. If we get to 1.2, we'll max out in July. If we get to 1.3, we'll max out in June. So again, it's sort of the inverse of what I showed you in Wuhan. When you take the distancing away, you're going to get the upswing because you don't have a vaccine and you don't have a treatment and you don't really have a good understanding of how many people are infected because your testing capacity is not adequate. A while back, I wrote a piece for the PSW website where I talked about some just various talking points, and I said you don't need to wear a mask in public because it's not really going to protect you from the virus. And I still believe that to be true. But the CDC and then Monica Gandhi and Open Forum Infectious Disease, they're making a really good case for why we should wear masks in public. They talk about the virus being contagious. It is. But, most adults that I see out in public wearing masks, they're taking them on, they're taking them off, they're not fitted properly. I don't think most people have a good understanding of how to use a mask. And as the weather gets warmer, people aren't going to want to wear them. They're going to want to take them off and take a breath, and that's just not appropriate. But CDC is recommending people wear masks when it's difficult to maintain 6 feet of social distance. And they specifically call out pharmacies. So it's something that you could talk to your patients about is when they're coming into the pharmacy and it's busy and they're waiting in line, they should consider a mask. And I think the most important thing that this does is it encourages people to maintain their distance because it's sort of a reminder that that person's wearing a mask and so is that person and I am. So we shouldn't all congregate less than 6 feet apart. So that I think is probably the best argument to wear a mask in public. And I think the second best argument is if you're asymptomatic, at least you're covering some of your droplets from going out into the world. But again, I don't think most people are wearing these things correctly. I want to talk briefly about vaccines. The global R&D for vaccines right now is remarkable. So it usually takes about 10 years to bring a vaccine to market. Ebola was on about a 5-year time course. Vaccines could be available under emergency use for the Department of Health and Human Services by early 2021. So that's about a year turnaround, give or take. There's already 5 studies in phase 1 trials. So as I alluded to earlier, you've got these 3 things we got to get into place to really control the virus and get our way of living back to the way it was pre-COVID, and that was a vaccine, treatment, and testing capacity. So vaccines are going to be crucial. But I'm gonna just put the disclaimer out here for all of you as pharmacists, as healthcare professionals. Given that this is going to be a rapid research and development, we have to be extremely cognizant of safety. There's already a lot of vaccine hesitancy in our communities, some more than others. If a safety signal is missed and whatever vaccine is brought to market causes harm, we're going to lose the public. And we're going to lose this option here for a COVID vaccine. So safety's got to be first and foremost as this accelerated vaccine development goes on. This is just another thing, another way to look at these data from Milken Institute, almost 200 treatments in consideration and over 100 vaccines in development. So again, the scope and speed of what's happening is unprecedented. I want to talk briefly about ACEs and ARBs. I talked last time about how there was this hypothetical harm with ACE or ARB therapy where the coronavirus would be more likely to bind and potentially cause harm, even though animal models didn't really substantiate that. There's been 5 observational studies now, 2 in China, 1 in Italy, 1 in the United Kingdom that was a preprint, and then 1 now in New England Journal from New York City in the United States. showing no harms in hospitalized patients with COVID-19 associated with ACE and ARB use. And there was 2 that had a signal of potential benefit, and they used multivariable regression analysis to show that there was a potential reduction in mortality in those who got ACEs or ARBs. And there's ongoing trials now to study these agents as therapy in patients who don't have hypertension or diabetes. So I've talked a little bit about the information overload, and I just want to bring to your attention something that's been coined by the World Health Organization as the infodemic, or an overabundance of information, some accurate, some not, that makes it hard for people to find trustworthy sources and reliable guidance. And this is where, again, I am urging you to use your voice as a pharmacist and a healthcare professional to dispel misinformation. There's so much scientific discovery and literature being published. A lot of it has not been peer-reviewed. We have to be skeptical. We have to appraise these data critically, look at the authors. Have they published before? Do the methods make sense? Do the results align with historical precedent? And the journalistic response to preprint data, as I mentioned, can be problematic. So we owe it to ourselves as a profession and to our patients and to our communities to be the gatekeepers of good information. So read as much as you can and appraise it critically. I keep coming back to Winston Churchill, who said, "A lie gets halfway around the world before the truth puts its boots on." And this is a really just telling graph. This is JAMA Internal Medicine. It was just a report submitted. This was internet searches for purchasing hydroxychloroquine. So trying to buy the drug hydroxychloroquine. Queries basically stable. Then Elon Musk endorses it. Trump endorses it, and it skyrockets. So people are believing this. It's getting a lot of traction in the lay press. And then, unfortunately, somebody was poisoned with chloroquine exposure, and now we've seen a downtick, but it's still not returned to normal. So we are really fighting 2 battles as pharmacists and healthcare professionals. One is to do right by our patients and secure the best treatment and advise the physicians treating these patients and, help those afflicted with the virus. And then we're also combating all this misinformation, which sometimes can feel like a much larger battle. I do want to leave you with this. This is a great perspective from New England Journal. And it was written by a physician or 2 physicians, and the audience is physicians. But basically they're saying, don't let your emotions take over your rationale or, your rational side. No specific therapy has yet to been scientifically proven to reduce mortality. And when you start focusing on all this minutia and the what ifs and the azithro and the hydroxychloroquine and the tocilizumab and all the stuff that Luke's going to talk about, that's important, but don't let it distract you from the core practices of offering the best quality supportive care. And if you want a good take on this, read Paul Sachs's blog here that I put on the upper right. He's an ID physician at Harvard, but he talks about the IDSA guidelines that were promulgated, and those guidelines essentially said, nothing can be recommended treatment-wise outside the context of a clinical trial. And he says that highlights the difficulty of don't just do something, stand there. So that's a play on don't just stand there, do something. Well, don't just do something. Sometimes you just have to stand there and wait for better data. And with that, I'm going to turn it over to Luke.
Thank you, Tom. I appreciate all of the work that's gone into this, and I think it's important to start with that Paul Sachs comment that this is a very challenging situation because we've all heard friends and colleagues talk about their experience. We have a few of our physicians that I work closely with who've trained in New York at some of these epicenters of care, and there is an absolute push to want to do something, and that makes it a very challenging place when we are as caregivers. We want to be able to give family members and patients something to hold on to, and actually the best thing to do may be to do nothing at this time. And I think that gets back to the idea of not really doing nothing, but focusing on the things that we already do well. We already have proven therapies and proven benefit associated with those. So I always start with this, is that it's focusing on and providing excellent care. And we talk about this in the context of critical care medicine, but this starts at the time that patients become infected or before they're infected. And Tom did an excellent job talking about the therapies, the quote unquote therapies we can take and recommend that include social distancing, masks, hand washing, and all of the things that we previously discussed. So I think that's an important place to start, that we do have something to offer. So start by educating your patients that we are doing something, and then focusing on the data that is there. Society of Critical Care Medicine has the surviving sepsis guidelines for COVID-19. Local institutions like UW, make our recommendations available to everybody. So making sure you're familiar with what to do to just appropriately take care of patients. So now we'll move into the edge of medicine, the edge of data and evidence right now. So let's start with remdesivir. Certainly getting the most exposure and interest right now, and it started with this report from the New England Journal of Medicine that was published on April 10th, and this was the compassionate use of remdesivir trial. So this is only 61 patients that received remdesivir. Average age was 64 years old. They were the thing to consider about this patient population was that these were critically ill patients. More than half of them were mechanically ventilated. Almost 10% of them were receiving extracorporeal membrane oxygenation or ECMO. And their primary goal was to see an improvement in the oxygen support class. So they ranked the need for oxygen support on a 6-point scale, and about 70% of patients saw an improvement in oxygen support class, which isn't dramatic, but it's something that we were hanging our hat on, saying, hey, look, there's an opportunity for improvement. Remember, this is non-controlled. There's not a placebo group. There's not a control group to measure what happens if we didn't give remdesivir. So we don't know if these 70% of patients would have improved without therapy or with therapy. 13% of patients died, and 60% of patients reported an adverse event, with 23% of those being a serious adverse event. Now, the serious adverse events, multi-system organ failure, septic shock, AKI, and hypotension, are common ailments and comorbidities associated with septic shock and the disease progression. So it's hard to tease these out from the drug itself versus the disease process. So April 10th, we have some exciting, interesting data. And now this is where it gets very interesting. 2 weeks later, April 29th, this is the article from The Lancet that was published. This is a placebo-controlled, randomized controlled trial. It was funded by the Chinese Academy of Medical Sciences. So it's for patients in Wuhan. They had planned to enroll 453 patients, and it was stopped early because they were struggling to enroll more patients after the quarantine measures had decreased the number of patients presenting to the hospital with COVID-19. So they had enrolled 236 patients. So this is something to consider, that what does this do to your power? This reduced the power, from 80% to 56%. So this we can't make statistical statements saying that there's superiority. We can only say, we can make some non-inferiority comments with this. The primary endpoint was time to clinical improvement. That's an important endpoint for us, as we want patients to get better. We want them to leave our ICU. We want them to not need mechanical ventilation, especially as we are looking to preserve those tools. Because this was a placebo-controlled trial, this was an overall less sick population than those patients who were receiving it for compassionate use. Challenges interpreting this data was that they also allowed the use of interferon, Kaletra, and corticosteroids. So we know that Kaletra had been studied in a placebo-controlled randomized controlled trial. It didn't seem to show any benefit, but there's still theoretical benefit that it may have an impact on COVID. The use of corticosteroids with 4 in 10 patients receiving corticosteroids was challenging because we don't know how to interpret that. Does that improve your mortality, or does it decrease your mortality, or increase your mortality? So some challenging confounders there. And ultimately, on the right, you can see the time to clinical improvement showed no difference between placebo and the control, or placebo and remdesivir. They did start highlighting some subgroup analysis saying that if you started remdesivir earlier, within 10 days of symptoms, you had a numerically faster time to clinical improvement, and there was some press that picked up on that. But this was really trying to stretch this data, looking at subgroup analysis and apply some of the tools that we learned from influenza treatment. And again, you'll notice 66% of patients reported an adverse event, and that was similar to those in the control group. So this gives us some idea that this is likely a safe drug. So that was published online at about 10 a.m. and 2 hours later, Gilead put out a press release announcing their top-line data from their NIH-funded study, which demonstrated similar efficacy between a 5 and 10-day dosing duration of remdesivir. This was paired with a statement about a second study that showed that you may be able to decrease time to clinical improvement administering remdesivir versus placebo. And about 30 minutes after that press release, Anthony Fauci was quoted in the news about remdesivir. So a lot of confusion, because you have a press release saying it works, the only peer-reviewed published study coming out saying there's no difference, and people were really left struggling. How do I make hay of this data? And to add to that challenge, the study that Anthony Fauci was talking about is a very complicated study. And it is referencing an adaptive study design. Now I want to take just a brief couple minutes to talk about what that is, because I think you'll be asking, people will be coming in and asking questions about this. So this is a study that's led by the NIH, and what you do in an adaptive study design is you start with 2 or 3 or 4 groups of patients. This study started with 2, a placebo and remdesivir. The idea would be that as new therapies become available, we add another arm. And at defined time points, there are interim analyses. And you can see the interim analyses time points here in the yellow box. So after a certain number of patients are enrolled and meet the endpoints, a data safety monitoring board reviews their outcomes and says, do we have enough? And is there a trend or a signal towards either safety or efficacy? If there's a safety signal, the trial's ended, or that arm is ended, and all patients are moved into the other group. So if alternatively, you could see an efficacy signal where, for example, remdesivir would be identified as being more effective than placebo. So instead of having all patients stay in the placebo arm, those patients are transferred to the remdesivir arm, and that becomes the new baseline. This allows us to add another layer, a new drug, a future drug, and compare it to remdesivir as the new standard of care. And this kind of iterative process allows patients to benefit the most from participating in a clinical trial, so that you don't have a clear indication of efficacy for one arm, and you keep patients in the placebo arm when they're not receiving a benefit. Alternatively, you may have a signal of harm And you continue in a traditional randomized controlled trial, you keep patients in that arm and continually expose them to that risk. So this is a way to adapt your clinical trials and help patients achieve the best outcomes while still adhering to a priori statistical analyses and outcome analysis. So the goal of this study was to start with an enrollment of 800 patients, and that included moderate, severe, and critically ill patients. And the statement that was included in the press release was that remdesivir had a 31% faster time to recovery than those who received placebo. And this was just highly statistically significant. And what that meant was that the remdesivir group had a time to recovery of 11 days versus 15 days for placebo. There was also a small mortality benefit, or at least a trend towards mortality in those patients receiving remdesivir, although that did not reach statistical significance. And this is obviously a subgroup analysis, so we can't put too much weight into that, and I would highly caution people in over-interpreting the mortality benefit at this point in time. So the study was, the placebo arm was terminated early, the study was terminated early, and they're completing the follow-up for patients that have been enrolled. They need to get to their 14-day endpoint and 28-day endpoint, and then the full study will be published, peer-reviewed, and ready for sharing. But in the meantime, that data has generated a lot of interest, and I think our leaders are still grasping for something to give to patients. So remdesivir was approved for emergency use authorization on May 1st. What that means is that it is now available to patients outside of clinical trials. And I'll remind everyone that the last time the emergency use authorization was used for an antiviral was the use of peramivir during H1N1. And ultimately, these drugs need to continue with phase 3 and phase 4 trials. And peramivir has shown very little benefit outside of that, after that emergency use authorization. So I caution people still about over-interpreting remdesivir. Early signals seem good, but there's still a lot to learn about this. Notably, the CEO of Gilead said that there was about 200,000 vials available, and those would be distributed through the US government. So I took a snapshot yesterday of the rate of infection. This is the new cases, new daily cases confirmed. And I know this is rather small, but you can see that over the last month, there's been about 25,000 cases confirmed per day in the United States. And if you apply some of the data that we know about how many patients meet the criteria for moderate, severe, and critical, and say those are the patients that might receive remdesivir, this means that we probably have 5 to 6 days, maybe 3 to 5 days of treatment available with this 200,000 vials across the United States. So that's great that we're going to have some drug available. There's not going to be as much available as we would like, and that distribution is going to come through the US government. I confirmed that today, that it will be distributed through AmerisourceBergen at the discretion of the US government. So it will target the hotspots, New York, Boston, and my prediction is that it'll be unlikely that Wisconsin pharmacies and hospitals will get remdesivir, get access to this first round of remdesivir. Now there are some reports that Gilead has 2 million more vials available, and I haven't seen confirmation of that, but once those become available, then I think we'll start to see access to remdesivir become more available here in the state of Wisconsin. So cautiously optimistic that remdesivir will be available, cautiously optimistic that it will be beneficial. I don't think that it will be available to Wisconsin patients for at least a week, maybe longer, depending on the supply that Gilead has in backup. So with that, I wanted to also address the hydroxychloroquine chloroquine status. many patients are still coming in and want to have something to do. The status of hydroxychloroquine and chloroquine, I think, has changed recently. Between the two, hydroxychloroquine is preferred, safer, a better safety profile. Supply chain has significantly improved. Almost universally, people are recommending this in the context of a clinical trial. And that's not because of any new data to show that there's harm associated with it. It's just a multitude of studies that have come out showing no benefit. Now, these studies are limited, they're retrospective, there's all the challenges associated with those. But I think what that does is shift our benefit or our risk benefit ratio toward risk, saying maybe that early benefit signal that we saw just isn't there. And we know that hydroxychloroquine can increase the QT, and early data showing that it increases it by about 25 milliseconds, which is equivalent to fluoroquinolones and voriconazole. And certainly caution needs to be given if you're going to, or taken if you're going to give it with azithromycin. Something that is available is convalescent plasma. This is a blood product. Oftentimes pharmacies aren't handling it or managing it, but it should be something you're experienced with and knowledgeable of. It's available through an expanded access program, which is very easy to enroll, 5 to 10 minutes for the physician at your local institution to get access to. There is a bank of plasma available, and patients can get enrolled into that. They're given 1 unit of convalescent plasma, which is about 250 milliliters of blood. They have to meet either one of these 2 criteria, severe or life-threatening, which you can see the inclusion criteria there. And from a monitoring standpoint, you're just looking for transfusion-related infection, so transfusion-related acute lung injury, and you may see some Tylenol and Benadryl given before and after. This, I think, speaks to this idea of a 2-phased process, right? The convalescent plasma and the antivirals are hitting the viral response phase, which is the early manifestation of the disease. And then as we move towards patients that are hospitalized and certainly critically ill, this is probably a host inflammatory response phase. Data seems to be just continuing to build, supporting this kind of 2-phase fashion. So these are things that you want to consider, is that if we can start remdesivir early or convalescent plasma early, the better. But once you move into this host inflammatory response phase, the targets are more interleukin cytokine mediated. And I took these tables from our guideline, and I think they just speak to the types of patients that you might be considering. One second here. So who might you consider for anti-cytokine therapy? These are patients that either already need invasive mechanical ventilation or patients with worsening O2 needs. So they have an increasing oxygen needs. They're usually tachypnic and are requiring O2 supplementation. And they have laboratory evidence of hyperinflammation. Our expert group took a stab at trying to define what that means, and we said you have to have either a CRP or a rising serum ferritin, one or both, plus another serum biomarker, either a lymphopenia, D-dimer, or an LDH greater than 400, to consider use of anti-cytokine therapy. And currently at UW, we're recommending tocilizumab or siltuximab. We prefer these to be done via clinical trial, but we recognize that providers may choose to administer this outside of a clinical trial to give patients their best opportunity. So contraindications, most importantly, are systemic infections, and then warnings and precautions are included there. We've evaluated a whole multitude of drugs. I don't have time to go through all of them, but I've included them in this table. The ones that have gotten a lot of interest are ivermectin and nitazoxanide. These will kill drugs or kill virus in a petri dish, in a cell line, but you can't give enough drug to a patient without experiencing toxicities. So we're not recommending those unless you're doing it by a clinical trial. And there's just not enough information about some of these other agents like anakinra or JAK kinase inhibitors to be outright seeking them for recommendations outside of a clinical trial. The other question that comes up is what to do with anticoagulation. And we get a lot of patients that, and reports recently of patients with a hypercoagulable state. We're still monitoring that data. Most of that data is still case series, case reports, and is subject to all of the limitations associated with a retrospective case series. So it's a non-controlled fashion. We're still recommending the use of anticoagulants in the context of a controlled clinical trial. So I guess to put it chronologically, patients will be admitted. They should be evaluated for the need for VTE prophylaxis. These patients will be high risk. If you're using the PADUO risk score, they have an infection, they're bedbound, and it's reasonable to treat these patients according to the VTE prophylaxis data that we know and has been tried and true and proven. So you can start these patients on low molecular weight heparin. If they have a DVT or a suspected DVT, it's reasonable to start them on therapeutic anticoagulation. We are not recommending alteplase outside of the clinical trial. And currently, we are not recommending the use of rivaroxaban or any of the DOACs at discharge outside of a clinical trial. We know that there's risks associated with these medications when it is in a clinical controlled setting, and we're concerned that the benefit may not be there if we start administering this to all patients. So I guess finally, I'd like to just wrap up, and I think this is a really important read, and I highly suggest people to just read through this. This is a publication that was written in 1919 after the 1918 influenza, and I think there's a lot of lessons to be learned. And the three factors that the author points out are that people do not associate influenza with the common cold. It's more discomforting than dangerous, and I think that they don't appreciate the risks that they run, and that there is a requirement for people to impose a restriction on their conduct, and more so than at the time was an enteric infection that they were comparing this to. So masks, social distancing, canceling of mass events, respiratory infections are unique with this, right? There's nothing like this with other types of infections, and the burden is both on the infected person, but also on the non-infected person. There's a societal component that this happens. So, I think the component to this that I found very interesting is this is published in May 30th. So it took months, and the author even says this is going to take months or even years to understand the full impact of this disease. So it's very challenging to understand the impact of COVID-19 and the mitigation strategies in real time, and you must look at it historically and in a historical context. And there's so many similarities to today's crisis, blaming foreign origin rather than looking locally, a desire to do something even when they recognize that it may be harmful, and a struggle to grasp the full impact of the disease. And I would highly, just in closing, I'd recommend listening to a medicine grand rounds that was recently presented by Dr. Judith Levitt. She's a medical historian. I've included the link here, and you can search for grand rounds. She gave a talk about the public response to smallpox in 1899 and 1947, and how vastly different the outcomes were. They talk about Milwaukee versus New York City, and the similarities between this and that the smallpox outbreak are just so stunning. There's so much to be learned from the historical context that these have happened in. So I would encourage you to check this out. It's a great listen to. She's a nice, easy, peaceful speaker to listen to. So highly recommend that as well. So with that, I know we're we're getting to the end of time, but I'm happy to stick around and answer lots of questions. So I know we have a bunch coming through. So thank you all for joining us. Thank you all for the work that you're doing to care for patients. And please let us know if there's any questions that we can answer.
Thank you so much. I do have a list of questions here that have come in on the question box. You're still welcome to enter more questions into the box, and I'll just add them to our list of questions, and we'll get to as many as we can between now and 1:30. I have also received some feedback that some people are having trouble with the question box. So if you are unable to use the question box, feel free to raise your hand on the use the raise hand function, and I'll try to unmute you. Or you can also try the chat function and chat me, Erica Martin, and you can enter in your question there. So any of those three options are great for asking a question, and I will try to monitor all three. So going to our first question. So if remdesivir data is weak or conflicting, why is there a push to use this medication?
Luke, do you want to take that one? Otherwise, I'm happy to opine. Well, I think there's probably a push to use this medication because we don't have anything else. I mean, it is the only agent thus far that in randomized controlled fashion has shown any semblance of benefit. I think another big reason that people want to use it is its safety profile is favorable. You know, hydroxychloroquine, azithro, they've got a lot of cardiac conduction abnormalities, QTc prolongation. People are hesitant with that. Tocilizumab, the interleukin-6 antagonist, that suppresses the immune system and can lead to exacerbation of chronic and acute infections. So there's hesitation with that drug. Remdesivir, from a safety standpoint, is much better. I think that's why people are pushing to use it.
Awesome. Thank you for that answer. I also forgot to announce that we will have the recording and the slides available. So I know if you have to hop off for a 1 o'clock meeting here, they will be posted on the PSW website within the next 24 hours. It does take a little bit of time for us to convert it to a file that can be on our website. So look tomorrow and you'll see a recording of this webinar on our website along with the slides. So with that, we'll continue with the questions. And Luke is having a little bit of trouble with his mic right now. So as he works that out, we'll go to another one. So could you comment on the link between Kowalski-like illness in COVID-19 positive children?
I cannot. I don't really have any experience with pediatrics and would not feel comfortable commenting on that. Sorry.
Right. That's all right.
This is Luke. Can you hear me now?
Yeah, we can hear you.
Perfect. There you are.
Sorry, I've been I will comment on just one brief thing. I've been sitting in with our pediatric work group as well. I would say that all of the data that's coming out about pediatrics right now is based off of case series and anecdotal evidence. So I would be cautious about over-interpreting this. There does appear to be some association with kids and an anti-inflammatory process that's going on as well. But the numbers are very challenging, and we need a national pediatric push. And I know there's some PIDs and SHARPs are trying to pull some of this data together. So I'd encourage you to get involved with a few of those to probably get better answers for that.
Thank you. All right, we'll move on to the next one. So can you discuss the link between smoking and contracting, spreading, and dying of COVID-19?
Can you repeat that question? There was a lot there, Erica.
Yeah. So can you discuss the link between smoking and then contracting, spreading, and dying of COVID-19?
Yeah, I mean, as best I can. Why don't I weigh in, and then I'll let Luke weigh in as well. In terms of contracting viral illnesses and smoking, there's definitely a link there. Smoking compromises your pulmonary parenchyma and your natural host defense mechanisms to viral and bacterial invasion in the respiratory tree. So those people are at higher risk. I think when you look at the data out of Italy, a lot of people in Italy do smoke tobacco, and they really had a hard time both contracting the infection and then with the outcomes. And I think when your respiratory tree is compromised through years of cigarette smoking, you are going to have a harder time recovering from a respiratory illness. If you get on the ventilator, it's going to be less likely that you're coming off the ventilator. So I think people who smoke and have long 5-year histories of smoking are more likely to get more likely to experience a worse outcome from this infection.
I would agree with Tom. The only other thing I would add is that there seems to also be a link with smoking and socioeconomic status as well. So there may be a component of overlap between those two that's contributing to worse outcomes.
Great. So our next question is, has any new data shown the typical time frame of this virus from contraction to resolution?
I'm happy to take the first stab at this one. So there does appear to be between 5 and 12 days from contracting the virus to showing signs and symptoms. And then patients who are, once you're hospitalized, these patients tend to be in the ICU for a long time, on the order of weeks. So 10 days, 14 days, up to 21 days. So the total resolution, from acquiring SARS-CoV to the end of your hospital stay can be up to a month, even longer. So these are very ill patients that stay ill for a long period of time.
Yeah, and I do think I have nothing to add to that. I just think it's important to reiterate that approximately 80% of people who contract this virus will have no symptoms. 20% will, and then 5 to 10%, give or take, are going to end up in the situation that Luke just described. So, the duration of the disease, if you will, kind of depends on the host, and we don't quite have a great understanding of why some people end up in the ICU for weeks and weeks, and some people never develop symptoms. It's such a novel virus.
Great. And we'll go to our next question. Is there any validity to the idea that masking otherwise healthy people may cause them to experience a decrease in immune functioning? That is, decreasing or normal interaction with bacteria and viruses will make us weaker in the long term for fighting otherwise benign infections.
Huh. Not aware of any data offhand. I mean, it's a reasonable hypothesis, but it's probably important to remember that you're not masking 24/7. And there's going to be periods where the mask is off, whether it's at nighttime or once you return to your home, etc. So I don't know. I think the benefits of masking, particularly like if you're talking about masking at work, if you're working in a hospital or a pharmacy or a clinic, far outweigh any potential hypothetical, detriments or harms.
And I would just throw in that unless you're wearing an N95 mask continuously, you're still being exposed to quite a few pathogens.
And our next question is, just under 3 million Americans pass away annually, largely from heart disease, cancer, chronic LRTIs, and other comorbidities such as suicide, hypertension, and assault. Is there a chance that these disease states may spike, I think, because of social distancing practice, possibly in excess of purported lives we saved from COVID, especially given that the estimate of 300 lives are saved in Wisconsin from safer at home?
Luke, I think when we were doing our initial discussions, you had some pretty good thoughts on the sort of flip side of focusing on COVID. I don't know if you want to take this one.
Sure. Yeah, I think this is, it's a very complicated answer. It's an excellent question. And I think there's a few ways to look at this. One of them being, yeah, we're going to see, probably fewer, fatalities from car accidents and shootings because people are at home. There may be a benefit to, the safer at home order. We see that already from the impact on influenza. The other thing I think about this is, this has been an unprecedented scenario where people are asked to learn about medicine and think about new things that they've not considered before. So there's a lot of people taking an interest in their health that maybe didn't take much of an interest before. So maybe they're learning more about the impact of diabetes or heart failure because of COVID. And I think that's an important thing to be ready for and to be ready to support. And I think they'll come to pharmacies and ask about that. And maybe we'll see a more medically literate population following from this. That's the optimist in me, is that I think people are understanding the scientific method, understanding experts, and we're seeing in real time, people predicting one thing, that it's, COVID's going to go away. Other people saying, no, this is, in my expert opinion, this is what it's going to do. And the data's there to see who was right and who was wrong in almost real time. I think that's also, bringing to light the challenge with this and the info overload, like Tom spoke to. A lot of people have access to a lot of information and also have a outlook and are a venue to write what they want to. People are out speaking and writing, things on LinkedIn and various sites on the internet. We need to be very cautious about who we listen to and how we identify experts. The pandemic isn't ripe for experts necessarily. Experts have a really deep amount of knowledge in a narrow spectrum, and a pandemic requires a deep amount of knowledge in a broad spectrum of things. So we're finding out who has that deep amount of knowledge. And I think people are hopefully learning that it's easy to mix data up and come up with the wrong prediction. And we have the data readily available now to see when those predictions are wrong. So I'm optimistic that people are learning about the scientific method and how to think about their health and take an interest in their health because of COVID. So.
Our next question is, for patients who are hospitalized and COVID-19 positive, is there data on how long these patients are contagious? Is it safe for them to be discharged into the community or back to a nursing home?
Well, generally it's thought that people are contagious for about 14 days. So when people are released back into the community after a hospitalization, they're usually put into some level of home quarantine to complete 14 days. I know a lot of nursing homes will say you were in the hospital, COVID positive, test or not, or no test, you're going to be quarantined for 14 days. So that's sort of the number that a lot of people cite. How it gets applied relative to symptom onset or treatment or discharge. Those policies are a little more heterogeneous, I would say. And it's not dissimilar from like how every LTAC and nursing home does, sort of like infection prevention and drug approvals and everything else like that. So it's something I've noticed anecdotally. There's no real consistent standard. I don't know what the experience has been elsewhere.
Hi guys, this is Sarah Sorum. I thought the point that was made earlier about this has created a situation where people are taking a larger interest in their health and thinking about the scientific method and other things like that. I think that's right on. We're hearing from experts that they're predicting that pharmacies will experience a 30% uptick in immunization requests in their pharmacies this fall, particularly related to more individuals getting the influenza vaccine. So that's a wonderful, wonderful thing, and will create challenges for pharmacies to address that. And I can talk at some point, if desired, about a little bit about some of the additional efforts we're doing around testing and the pharmacist's role in testing. But I wanted to circle back on the point about sort of this ethical dilemma and also the feeling of, if we feel like we're overreacting, we're doing the right thing. And maybe if you could comment a little bit about, do you feel that where Wisconsin's at currently, we've overreacted? I know I've heard from health systems that are furloughing staff, et cetera, because of their, decreasing their census to be able to take on surge capacity, et cetera. What are your thoughts on that?
I'll jump in here. So I do not think we've overreacted. And I, as an ID expert and somebody who's critical care trained, I read and I saw, and I've been thinking about this, and I think this anecdote kind of explains my thoughts on this, is that we see patients come in with sepsis and septic shock all the time, and we administer antibiotics very early. And most of those patients get better and go home. And nobody's ever once said, man, I wish I didn't start those antibiotics earlier and just rode this out and seeing what happened. So I think that's an important thing to look at, right? We took our treatment, our social distancing, and we applied it to our society. And we're seeing the benefits of that. Patients aren't getting sick. COVID's not spreading as rapidly as it was before. And I think that's an important perspective to share with patients, is that we took our treatment, our social distancing, our changing of our societal norms and some inconvenience that we've had to accept, and we're now seeing the benefit of it. And I can't think we can't say that what we did was an overreaction. We got exactly what we expected, was an improvement and a low number of patients that wound up needing our care. It's unfortunate that we're gonna still struggle to quote unquote pay for that treatment, right? With furloughs, hospitals struggling to pay bills, but that's exactly what we would do for any other disease state, is start early treatment and expect to see positive outcomes. And we did that. And now we need the help from PSW to and as a group, Wisconsin, as pharmacists, we're gonna have to pull together and figure out how to work together to get through some challenging times as we pay for that treatment, with furloughs and smaller workforces for a time being. So I think that's something that I think we need to be ready for as a pharmacy profession, and certainly in the pharmacy profession in the state of Wisconsin. So I appreciate the work that PSW is doing, and I'm excited to hear all the things that are going to be available from PSW to help pharmacists work together.
Yeah, Luke, this is Sarah again. I couldn't agree more in terms of, that measure treatment approach. I think that's such a great way to put it. So thanks for that. I will say that this has created such a change for everybody in their workplaces, and thinking about now, how do we take some of these rapid changes that had to occur and move into what's next and the opportunities that presents, the challenges that presents. And I'm just so inspired by the people that we have to have those conversations about what that looks like. So we're hearing unique challenges from all different practice settings and also unique opportunities that now we're able to open the door on those conversations where those couldn't exist previously. So with that, I'd say, PSW is thinking strategically. We're thinking beyond just the short term, which the short term is extremely important, but thinking about long term, what does this mean a year from now for practice? What does this mean 5 years from now? And also, how can we use this crisis situation to build support for some of the things that we've presented as opportunities for quite some time? Pharmacist payment for services, expanded service offerings in community pharmacies, expanded ways to utilize pharmacists in health systems, expanded ways for pharmacists to care for populations of patients. So I'm done opining for now, but I really appreciate the question and the thought.
All right. Well, thanks. Well, that kind of answered. Actually, we had another question or two on that about related to what happens for long term with expanded services and supporting pharmacies continuing to offer services. So I believe that answered that question. We have two kind of about how the COVID-19 spreads. So the first question is, in regards to social distancing, outside of the data from China, what other evidence supports social distancing versus herd immunity? How does this compare with the results coming from Sweden?
Well, I think when you talk about herd immunity, the virus is so new, it's hard to, you look at other viral infections and you can make a case for herd immunity, and that's definitely going to be a long-term boon for our armamentarium against COVID, whether it's be a natural infection or vaccine. We're going to need community protection. I think the case of some of the countries like Sweden, and even when you look at South Korea and how they've gone about controlling the virus differently than we have in the States. I think there's just social norm differences between those countries and here. They're definitely more of a collective mentality. We have a much more sort of individualistic mentality. So I do think it makes it hard to achieve some of the gains that they have, particularly in Sweden. But the virus is highly transmittable, whether it's droplet or airborne, and the way this country works and the way that we think and the way that we behave, absent a vaccine, I'm firmly in the camp that social distancing is what we need to do to mitigate the damage of this virus. And that's all I'm going to say about that. Oh.
And
my dog apparently wants to say something.
Our last question for today is, are we seeing spread of COVID-19 in daycare workers like seen in nursing homes, as daycares are sometimes open currently under safer at home?
I haven't seen data on that. I would have to look that up. I haven't seen that.
I've not seen that data either. So I do not know.
The one thing I will say, though, when you talk about other reservoirs per se, prisons are a big reservoir right now. That's something that I don't think either one of us talked about. That's a that's a big problem right now in the United States is how do you mitigate the spread of SARS-CoV-2 in the incarcerated population?
Well, thank you so much for answering all those questions. There's one more that came in, and since we do have a few more minutes, I will quickly read this last one here, and then any others that trickle in, I'll write them down, and we'll either include them in a future webinar or on the PSW website. But again, they're all being recorded. Your questions are being recorded, this webinar is being recorded, and the slides will be posted on our website within the next 24 hours. So the final question is, with this being highly contagious respiratory spread, with what we have seen in cruise ships, any thoughts for hotel rooms and their ventilation systems, somewhat like Legionnaires?
Legionnaires is sort of different because, you know, it has this proclivity to sort of like embed itself in water and reside on surfaces and things like that. I don't think we've got a great understanding on SARS-CoV-2's propensity to reside on surfaces. It's definitely not spread through water or anything like that. So I don't think it has the potential to be spread like Legionnaires is, but I also can't really deny it. I don't know if anyone's looked at that in detail. We're still really trying to understand this virus.
Yeah, I'd say there's some differences between Legionnaires and SARS-CoV. Most importantly, probably, is the longevity of Legionnaires. Right, Legionnaires can survive in an air conditioning system with access to water for quite some time, and you know, SARS-CoV can live in for several days, but it doesn't replicate well. It's a very different pathway than Legionnaires. So I agree. I don't think we know enough about the transmission in specific scenarios like that, but it would still be a similar scenario, you know, where you could mitigate it by hand washing, masks, not necessarily that you need to wear them all the time in there, but you know, hand washing, masks, good general hygiene practices as well should be used.
Well, thank you so much for all those questions. I'll turn it over to Sarah to provide some closing comments.
Thanks, Erica. And I just want to say thanks again to Luke and Tom for all you've done to put together this wonderful material and share it with our members. Erica alluded to our website that continues to grow with information about pharmacy practice considerations related to COVID-19, and so I'd encourage folks to check that out. There continues to be activity in the regulatory waiver space, so we're trying to keep people updated about that. So the best vehicle for finding out that information is through Friday's Fast Facts communication, as well as that website. As it relates to testing, this has been a very hot area for a number of our members in community practice. There'll be continued information. Again, Fast Facts weekly will be a great source of that information. We remain very concerned about a few systems changes that need to occur that Luke and Tom referenced earlier in the webinar. So we're working on that very actively and have direct lines of communication with the State Emergency Operations Center, Wisconsin Medicaid, the Division of Public Health, and others, the governor's office too. So we're working really hard on your behalf. We know that now more than ever, pharmacists need advocates and pharmacy professionals need advocates. The other critical space is we're approaching graduation time, and I know we have a number of student pharmacists joining us on today's call, and there's that's of high importance to us, is to provide as seamless as possible the transition for our new graduates out into practice. So we are working on that and are happy to field questions and concerns throughout the weeks here with your emails to our staff. So thanks again to everyone, and we hope people stay safe and take care of your families, yourselves, take care of yourselves as whole people, and thanks for all you're doing for patients.
Okay. Thanks all.